Welcome to Simply Biology with Manisha. In this series, I am explaining the process of DNA isolation. In part 1, I had talked about the types of DNA isolation procedures. I had also discussed how cells are lysed in order to prepare a cell extract. Those of you who have not watched the part 1 video, please do take a look at it for better understanding of the whole process. In part 2, I am going to explain the methods of DNA purification. We have seen in part 1 that the cell extract contains DNA, RNA, proteins and several other biochemicals. In step C, the contaminants are removed so that pure DNA solution remains in the extract. While purifying DNA, certain reagents are added to the cell extract to degrade the contaminants. The simplest of these methods is organic extraction, in which phenol or a combination of phenol and chloroform is used. This is followed by enzyme digestion using the enzymes protease and ribonuclease. The standard method of removing contaminants is organic extraction. First, an organic reagent like phenol or a combination of phenol and chloroform in 1:1 ratio is gently mixed with the cell extract. After centrifugation, aqueous and organic solutions get separated. In between these two layers, you can see a sort of boundary layer commonly called the interface. The precipitated protein molecules can be seen as a white colored coagulated mass at this interface. In this figure, for clarity, I have shown the interface using green color. The upper aqueous solution contains the nucleic acids, that is, DNA and RNA that were present in the cell extract. Using a pipette, the aqueous solution is transferred to a fresh tube. The precaution to be taken here is you have to ensure that the pipette tip does not touch the interface. Otherwise, the precipitated proteins will again contaminate the aqueous solution. Often, the cell extract has a very high protein content, and a single phenol extraction is insufficient to remove all the contaminants. The first solution to this problem that one can think of is to repeatedly treat the cell extract with phenol, or in other words, repeatedly add phenol, centrifuge and pipette out the aqueous layer, and you continue the process until you get a clear interface, which means that all the protein has been removed from the solution. But this method is generally not used. The reason being that with every mixing and centrifugation step, a small fraction of the DNA molecules undergoes breakage. So at this point, a question arises: how to remove this protein contamination? The answer to this question is enzyme digestion. Prior to phenol extraction, a protease like pronase or proteinase K is added to the cell extract. The protease breaks down the polypeptides into smaller fragments. Now, once you add phenol and centrifuge the mixture, these fragmented peptides very easily settle down at the interface. And the aqueous solution contains both the nucleic acids, DNA and RNA. Now again, the next problem is we want only DNA to be present in the solution. So how to remove this RNA? Yes, you guessed right. Again, the answer is enzyme digestion. A few of the messenger RNA or mRNA molecules are removed by phenol treatment itself. But to completely remove the RNA, the most effective method is treatment of the cell extract with ribonuclease. Just like protease treatment, ribonuclease treatment is also performed before extraction with phenol. Ribonuclease degrades the RNA molecules into small ribonucleotide subunits. And finally, after phenol extraction, we get pure DNA in the aqueous solution. Whether it is organic extraction or enzyme digestion, both the methods are slightly laborious. A slightly faster method is ion exchange chromatography. Here, an anion exchange resin is used. This method makes use of the different electrical charges that are present on different molecules to separate these molecules from a mixture. In ion exchange chromatography, a chromatographic matrix or resin is used, whose particles are electrically charged. You already know that the nucleic acids DNA and RNA are negatively charged because they contain a negatively charged phosphate group, which helps them in binding to the positively charged resin. Now, the presence of salt interferes with this electrostatic attachment. Depending on how tightly the nucleic acid molecule is bound to the resin, the salt concentration needed to break this attachment varies. By gradually increasing the salt concentration, you can separate different types of molecules one after another from the resin. In the first step of this procedure, we take a tube made of glass or plastic and insert a resin into it, basically forming a column of resin inside the tube. The resin that is used is a positively charged molecule called diethyl amino ethyl cellulose, in short DEAE. The cell extract is added on top of this column. This cell extract does not contain any salt. So, all the negatively charged molecules bind to the resin. Now, when we add salt solution of very low concentration to the column, the first molecules whose attachment with resin breaks are proteins and they are eluted from the column. Next, if we add salt solution of slightly higher concentration, then RNA molecules are eluted from the column. And when highly concentrated salt solution is added to the column, at last DNA is eluted. This means that just by changing the salt concentration, you can elute different types of molecules through the anion exchange column in a single extraction. Generally, the eluate trickles down from the column because of gravitational effect, from where it can be collected by placing a tube under the column. However, to speed up this process, the column is inserted in a tube and centrifuged. Hence, it is called the spin column method. Overall, two factors, pH and salt concentration, control the binding and elution of DNA from an anion exchange column. This method has two advantages. The first one is that very pure DNA can be extracted by using this method. And the second advantage is that the resin can be renatured and used repeatedly. The only disadvantage of using this method is that while elution of the DNA, we use salt solution having very high concentration, which makes the extracted DNA unsuitable for downstream applications. Thus, the DNA needs to be desalted. Otherwise, the excess salt interferes with the downstream procedures. There is another efficient method of DNA extraction where we use silica and guanidinium thiocyanate. In alkaline conditions and presence of high salt concentration, silicates show high binding affinity with DNA. On the other hand, guanidinium thiocyanate, in short GUSCN, is a chaotropic agent, which means that hydrogen bonds break in the presence of guanidinium thiocyanate. For example, water loses its hydrogen bonds in the presence of guanidinium thiocyanate. It is a known fact that water is present inside as well as outside the cell. And we also know that hydrogen bonds in water molecules play a huge role in functions like the maintenance of structure of cell membranes or the suspension of proteins and other biochemicals in the cytoplasm. Since guanidinium thiocyanate disrupts these hydrogen bonds, it is very commonly used for animal cell lysis as well. Can you guess why? The reason is that animal cells do not have cell wall. So, guanidinium thiocyanate directly disintegrates the cell membrane. The second advantage of using a chaotropic agent like guanidinium thiocyanate is that in its presence, cellular proteins, For example, nucleases get inactivated. Thus, guanidinium thiocyanate is used both for DNA and RNA isolation. While purifying DNA, silica and guanidinium thiocyanate are used in three ways. First, in column chromatography, second, by centrifugation, and third, by using magnetic beads. Now, I will explain these methods in detail. In the first method, we use column chromatography. But here, unlike ion exchange chromatography, we use silica column instead of resin. Silica can be used in the form of gel or as glass microfibers. Another difference in this procedure in comparison with ion exchange chromatography is that the cell extract is mixed with guanidinium thiocyanate before adding to the top of the column. In the presence of guanidinium thiocyanate, the negatively charged nucleic acids present in the cell extract selectively bind to the silica matrix. Proteins and other biochemicals are unable to bind to the silica matrix and are eluted immediately. In this technique, sodium ions play a very important role. You must be wondering how. These sodium ions act like a cation bridge. Just imagine that there is a cationic bridge made of sodium ions which are connected to the phosphate backbone of nucleic acids on one side, and on the other side the silica matrix is present. Two conditions are important for the formation of this attachment. First, high salt concentration, and second, acidic pH, because of which this phenomenon is also called as salting out of nucleic acids. In other words, under high salt conditions, nucleic acids become insoluble and attached to the silica matrix, and this phenomenon is called salting out. To break the attachment of DNA with silica matrix, we need hyposmotic solution and alkaline pH conditions. In this way, DNA gets eluted. Now, take a look at this flowchart. After elution of the contaminants, DNA is eluted using a hyposmotic solution like nucleus-free water or a buffer like alkaline tris EDTA. Through this technique, we can obtain extremely pure DNA. This procedure is very easy to perform, and if you repeat the experiment, the results are reproducible both qualitatively and quantitatively. The disadvantage of using this technique is that small DNA fragments get very tightly bound to the silica matrix and thus are not efficiently eluted from the column. In the next method that I'll explain to you, this problem gets solved. And to know about that method, please subscribe to my channel for the next video upload. So, in part 3 of this video series, I will discuss some more methods of DNA purification and concentration. Let me ask you one question now. What kind of molecules are precipitated when you add phenol to the cell extract? DNA, RNA, or proteins? Type your answer in the comment box below. Please like this video and subscribe to my channel Simply Biology with Manisha. Thanks for watching.